just had a very good question someone in the class asked. They asked if the lab reports on your group experiments should be single-spaced like it is in the example that I put on Canvas, or whether it should be double-spaced. The example that I put on Canvas was actually single-spaced because it was formatted exactly the way that it is published in print. However, in most educational situations, instructors prefer that you double space. Isn't that usually the case when you submit a paper? Don't your professors usually want you to be double spaced? Yeah. The reason for that is it's easier to read, and it provides more room to make comments. If you single space, you can't. There's really not much room to make a comment in the middle of a paragraph or a sentence. So actually, even though it's supposed to be single spaced, I would prefer if you double spaced it. Okay, it just makes it easier for me to grade. And then, if you really were to publish it, it's really easy to switch from double spacing to single space. So if you would double space your paper, I would appreciate it. Okay? And you do not have to put it in a 2-column per page format like the example is either. You can just do it 1 single column per page. That makes it easier to read too. So, okay? Any other questions about the write-up? Okay, the instructions we went over during one of our labs, and we had a quiz over the instructions. So there was a lab assignment. It wasn't the quiz, it was an assignment. And if you click on the assignment in the instructions, is where the instructions for the lab report are. Okay. I would just kind of look at the example, and you want to put things in the same format, like the tables or graphs. You want the same format as in that paper. You want the same order of sections. Generally, the standard order is the title, then the author's names and addresses, then the abstract. You can skip the new finding section. Most journals don't have that section, so you can skip that one. The abstract, then introduction, then methods, results, discussion, and then references. They call it references in that journal. Most journals call it literature cited because that's really what it is. It's a list of the references that are cited. So I got a review question up here for first, if you would take a look at that and answer this to start out with, and we'll move on, and we're going to talk about gas transport in blood today. Missing a few people today. Where's everybody that usually sits up here? First 2 rows over here. Okay, let's take a look at your responses. Almost all of you said all of the above, and that is correct. So all of those things are factors that affect the rate of gas exchange. So as I said, today we're going to take a look at the transport of respiratory gases in the blood. So the next step after gas exchange between the air in the lungs and the pulmonary blood is the transport of those gases from the lungs to the tissues of the body. And both CO2 and oxygen are transported by the blood. Transport occurs both in the plasma as well as by the red blood cells. but red blood cells play an important role in the transport of both oxygen and CO2. Okay? So as it turns out, there are just two mechanisms that are used in transporting oxygen in the blood. Carbon dioxide, however, is a little bit more complicated because there's actually three different mechanisms that are involved in the transport of carbon dioxide. Okay. So we'll take a look at the transport of oxygen first. Oxygen is not very soluble in water, and plasma is made up of mostly water. But because of the relative insolubility of oxygen, only about 1.5% of the oxygen that's transported by the blood is transported as just dissolved oxygen in the plasma. So about 98.5% of oxygen is actually transported by the red blood cells. And the part of the red blood cells that oxygen binds to is the heme group that's found in the hemoglobin protein. Okay, basically red blood cells are not much more than just a bag of hemoglobin. They don't have a nucleus. They don't have many other. Cellular organelles, but they have lots and lots of hemoglobin molecules. And this is a model of hemoglobin up here on the upper right. And there are 4 different subunits or globular proteins that make up the whole hemoglobin molecule. Each one of those subunits contains a heme group, which is shown on the lower right. And if you look right in the very center of the heme group, what atom do you see? What element do you see right in the middle of that heme group? Or you can just tell me the abbreviation for it. What does that say? Right in the middle. It's small, but some of you should be able, right there, right in the middle. Iron. It's iron, right. There's an iron atom right in the very center of the heme group. So this is the heme group right here. This is the whole hemoglobin molecule up here. So there are 4 different subunits, and these heme groups are shown up here in red. So you see these 4 reddish colored figures. Each one of those is a heme group. Each heme group can accept 1 oxygen molecule. So oxygen comes in the form of a diatomic molecule. It's 2 oxygen molecules that are bonded together. So each hemoglobin molecule then can carry a maximum total of 4 oxygen molecules. Okay, so if you have 100 hemoglobin molecules, those 100 hemoglobin molecules can transport a maximum total of 400 oxygen molecules. Okay. However, the exact number of oxygen molecules that any given hemoglobin molecule is carrying depends upon the partial pressure of oxygen that hemoglobin molecule is exposed to. If you have a very, very low oxygen concentration, hemoglobin's not going to be completely saturated. If you have a real low oxygen concentration, each hemoglobin molecule only may be carrying 1 or 2 oxygen molecules. But at the partial pressures of oxygen in the lungs, usually oxygen is 100% saturated. So each hemoglobin molecule is carrying a total of 4 oxygen molecules. So transport of oxygen is really pretty simple. It just binds to hemoglobin and it's carried by hemoglobin to our tissues where the oxygen diffuses away from the hemoglobin and into our cells. Okay? Now, when oxygen binds to hemoglobin, it forms a compound called oxyhemoglobin. So oxyhemoglobin is what hemoglobin is called when it's combined with oxygen. Okay, as long as one oxygen molecule is bound to hemoglobin, it's called oxyhemoglobin. If the hemoglobin molecule is not carrying any oxygen, like it might be after it has passed through the systemic capillaries and given up all of its oxygen, then it's called deoxyhemoglobin. So deoxyhemoglobin is just the hemoglobin minus any oxygen. Now, when hemoglobin gives up its oxygen to our tissues, it also loads up with carbon dioxide, because oxygen and carbon dioxide transported in opposite directions. And so when hemoglobin is loaded up with carbon dioxide, it's called carbaminohemoglobin. Okay, so that's hemoglobin that's carrying carbon dioxide. So those are just some different terms that physiologists use to refer to different forms of hemoglobin. Now, one interesting characteristic of hemoglobin is that it exhibits what's called cooperative binding to oxygen. So when oxygen first starts to load up onto hemoglobin, when oxygen first starts to load up on hemoglobin, at low partial pressures of oxygen, the percent saturation of hemoglobin is very low. Normally, the partial pressure of oxygen in systemic tissues is fairly low. It's only about 40 millimeters mercury. So at those low partial pressures of oxygen, the saturation of hemoglobin is about 79%. Okay. If you increase the partial pressure to, from 0 to 20, the saturation of hemoglobin increases very, very rapidly. But at higher partial pressures, the hemoglobin molecule binds oxygen less tightly. Okay, so if you do a graph of the percent saturation of hemoglobin versus the partial pressure of oxygen, the shape of the graph is said to be sigmoidal in shape. It's kind of an S-shaped curve. Okay, so at real low partial pressures, hemoglobin doesn't bind oxygen very tightly at all. At high partial pressures of oxygen, hemoglobin binds oxygen very, very tightly. Okay? And if you really think about it, it kind of makes sense, because the place where the partial pressure of oxygen are highest are in the lungs. And the lungs is where hemoglobin loads up on oxygen. So you wouldn't want hemoglobin to unload its oxygen right away as soon as it gets loaded up in the lungs, because that's not where the oxygen is needed. But at very low partial pressures of oxygen, like in our systemic capillaries, hemoglobin doesn't bind oxygen very tightly. And so oxygen unloads very easily. And that's exactly the place where you want oxygen to unload. So hemoglobin holds oxygen differently in different parts of the body. It doesn't bind oxygen very tightly at all, when the partial pressure is low, like it is in the systemic capillaries, but it binds oxygen very tightly in the lungs where you want the hemoglobin to load up with oxygen. So hemoglobin is adapted very, very well for the transport of oxygen. Okay? So this is called a sigmoidal oxyhemoglobin dissociation curve. Okay, so as blood flows to our tissues and the partial pressure of oxygen starts to fall, the percent saturation of the hemoglobin drops off very, very rapidly, because that's exactly where you want it to deliver the oxygen. All right. So that's called a sigmoidal curve. So this is a diagram that is showing the delivery of oxygen by the blood to our systemic tissues. So over here on the left, this yellow shaded area, that represents the cells of our body. Okay, you can sort of, if you look closely, you can sort of see the outline of some small cells. Over here on the right is a red blood cell that's located within a capillary. So this whole pink shaded area is inside the capillary. Over here are individual cells. Okay. Now, as it turns out, carbon dioxide is transported in 3 different ways by the blood. Okay? So carbon dioxide is a lot more soluble in water than oxygen. So carbon dioxide can just diffuse directly into the plasma of the blood fairly easily and in larger quantities because it's more soluble. It dissolves in water more easily. So about 7% of all the carbon dioxide that's transported by the blood is just transported by dissolving directly in the plasma. It's transported as dissolved CO2 in water. And that water is in the plasma. Now, another 23% of the carbon dioxide, so almost a fourth of the carbon dioxide, is transported by binding to hemoglobin, but not the same part of hemoglobin that oxygen binds to. Okay, oxygen binds to the heme group. Carbon dioxide does not bind to the heme group. Instead, carbon dioxide binds to the globin portion of the hemoglobin. It binds to the subunits of the protein, okay, the polypeptide chains. So 23% of the CO2 binds to the protein portion of hemoglobin. 7% is just transported by dissolving in the plasma. So 23 plus 7, that's 30%. So almost a third. That's how about a third of the CO2 is transported. That leaves another 70% to be transported by a third mechanism. So about 70% of the CO2 diffuses out of the tissues of our body through the capillary walls. And once it gets into the bloodstream, it diffuses into the red blood cells where it combines with water. And inside of red blood cells is an enzyme, which you can see right here. It's abbreviated as CAH. CAH stands for carbonic anhydrase. It's an enzyme that catalyzes the reaction between carbon dioxide and water. When carbon dioxide and water unite, it forms a weak acid called carbonic acid, which is shown right here. Carbonic acid's formula is H2CO3. Okay? So that's a weak acid, which means some of those carbonic acid molecules will give up a hydrogen ion. to the surrounding fluid inside the red blood cell. And so when carbonic acid gives off a hydrogen ion, it forms a bicarbonate anion. Bicarbonate is CO3, which has a negative charge. And so again, CO2 diffuses into the red blood cells, it combines with water, and in the presence of carbonic anhydrase, it forms carbonic acid, which gives off a hydrogen ion to form bicarbonate, plus a hydrogen ion. Now, that bicarbonate anion, it's called an anion because it's negatively charged, it is exchanged for chloride with the plasma. So what happens is there's an antiport transporter protein in the cell membrane of the red blood cells, and this transport protein transports bicarbonate out of the red blood cell, and in order to maintain electrical neutrality inside the red blood cell, it exchanges the bicarbonate for a chloride ion. Chloride is also negatively charged, just like bicarbonate is negatively charged. So bicarbonate moves out of the plasma, and then chloride moves into the red blood cell. The only purpose, well, I should say it's the only purpose. One purpose of this exchange of bicarbonate for chloride is to maintain electrical neutrality across the membrane of the red blood cells. But there's a big advantage to doing that. The big advantage of moving the bicarbonate out of the plasma is it reduces the concentration of bicarbonate on the inside of that red blood cell. And by reducing the concentration of bicarbonate in the red blood cell, it shifts the equilibrium of that equation to the right. So it makes it easier for water to combine with carbon dioxide to form carbonic acid, and it makes it easier for the carbonic acid to dissociate into bicarbonate and hydrogen ions. And so what that does, it allows the blood to transport more CO2 in the form of bicarbonate. So about 70% of the carbon dioxide produced by our tissues is transported by our blood as bicarbonate ions. Okay? So that process of forming bicarbonate from CO2 starts in the red blood cells. Once the bicarbonate is formed, it's transported into the plasma. By getting it out of the red blood cells into the plasma, that shifts the equilibrium of that equation to the right. It allows more bicarbonate to be formed. And so more CO2 can be transported as bicarbonate. So the majority of your CO2 is transported in the blood as the bicarbonate ion, this CO3 that's negatively charged. About 23% is transported by binding to the globin portion of the hemoglobin. That's the protein portion of the hemoglobin, not the heme group. And another 7% is transported just by dissolving in the plasma. Okay? So this is what happens into CO2 in the systemic capillaries surrounding our tissues. Now, down here it also shows how oxygen is transferred from the red blood cells to the tissues. So 98.5% of it was bound and transported by the heme group of hemoglobin. So the oxyhemoglobin is converted into deoxyhemoglobin when the hemoglobin gives up oxygen to our cells. And then just a very small amount, about 1.5%, just diffuses from the plasma into the cells. But the majority of the oxygen is transported by binding to hemoglobin. Okay. So this is a diagram that's showing what happens once the blood arrives at our tissues, after passing through all of the arteries in our body. Okay. Now let's look at what happens when that blood, which has given up its oxygen and it has picked up a lot of carbon dioxide, it's transporting lots of carbon dioxide as bicarbonate, when it gets to the lungs. Okay, so on this diagram, on the right-hand side is the inside of a pulmonary capillary that surrounds the alveolar sacs. And over here on the left is the air that's in the alveoli. So that's the inside of all the little tiny air sacs. Okay? So when the blood gets to the lungs, what happens is chloride ions are transported out of the red blood cells. Remember, those moved in when bicarbonate moved out in the tissues. So chloride is transported out. When that happens, that causes bicarbonate to be transported back into the red blood cells. That increases the concentration of bicarbonate in the red blood cells. So that shifts the equilibrium of that equation back to the left instead of the right. So the more bicarbonate that loads up in the red blood cells, the more bicarbonate will combine, the more tendency it will have to combine with hydrogen ions to reform carbonic acid. And once the concentration of carbonic acid increases, that shifts the equilibrium back to the left because this is a reversible reaction. And carbonic anhydrase can also catalyze that reaction in reverse. And so the carbonic acid will be converted back into CO2 and water. Okay? When the CO2 concentration rises, then the CO2 then diffuses from this area higher concentration to lower concentration inside the alveoli. And so that causes CO2 to be unloaded from the red blood cells into the alveoli. Some of that was also transported by being bound to the globin portion of hemoglobin. And so the CO2 will disassociate from hemoglobin and diffuse from the area higher concentration to lower concentration. So that accounts for 23% of the CO2 that's transported. And likewise, that 7% that was transported just by dissolving the plasma, that also will diffuse to this area of lower concentration in the alveolar spaces to transport another 7% of the CO2 out of the blood and into the alveoli. And then once the CO2 is transported into the alveoli or the alveolar sacs, then it can be exhaled by our respiratory system during expiration. Now let's take a look at what happens to oxygen when the blood reaches the lungs. So because this venous blood that's returned from the lungs has given up most of its oxygen to our tissues, the concentration of oxygen in the venous blood, once it reaches the lungs, is very low. And so oxygen will move from an area higher concentration in the alveolar air into the red blood cells, where most of it binds to the heme group of hemoglobin. So that's how 98.5% of the oxygen loads up into the blood is by binding to hemoglobin in the red blood cells. A very small amount, about 1.5%, dissolves directly in the plasma. And that's because oxygen is not very soluble in water. Okay. So oxygen is moving out of the alveolar spaces into the blood, and CO2 is moving in the opposite direction. Okay, so this diagram as well as the previous one, this is kind of showing what happens at the end of gas transport and what leads to gas exchange, which we looked at in the previous section of our textbook. Okay? Any questions? Now, there is a little bit of chemistry involved in gas transport by the blood, but really there's only the only complicated thing involves only one reversible reaction, and that's the combining of CO2 and water to form carbonic acid, which gives off a hydrogen ion to form bicarbonate. And that's actually how most of the CO2 is transported, by converting it to bicarbonate. All right. Now that by the way, as we're going to see later, that bicarbonate that's produced due to the transport of carbon dioxide by the blood, since that's negatively charged, bicarbonate actually acts as a pH buffer in the blood, because bicarbonate can combine with hydrogen ions to form carbonic acid, thereby neutralizing hydrogen ions. So bicarbonate is a really important pH buffer in our body. So it sort of serves two purposes. It's a way of transporting CO2 in the blood. It's also a way of preventing the pH of our blood from getting too low. Okay, because if the pH of our blood would get too low, all of our other body fluids the pH would get too low. If our pH gets too low, that can disrupt the function of enzymes, which can cause lots of problems. All right. Now, another interesting thing about hemoglobin is that its affinity for oxygen can change depending on the chemical environment of our body. So when our body is very metabolically active, that's when it needs the most oxygen. And so there's several signs that there's several chemical and physical changes that occur when our body is in need of more oxygen, because it's using up more, it's using up more ATP, and it needs to synthesize more ATP, and so it needs more oxygen for aerobic respiration. Some of those signals that our tissues are being more busier, doing metabolically active, is higher temperature, because contracting muscles generate heat. So higher temperatures mean that there's probably a need for more oxygen. Okay. Also, metabolically active tissues are produce lactic acid from anaerobic metabolism. Also, more CO2 is produced. Okay. More CO2 is produced by Krebs cycle when tissues are metabolically active. And a compound called bisphosphoglycerate. Bisphosphoglycerate is actually one of the intermediate compounds in glycolysis, which is the first pathway involved in ATP production. So if any one of these chemicals or heat increases, that's a sign that there's a greater demand for oxygen. And as it turns out, all of those factors will cause hemoglobin to decrease its affinity for oxygen, which means that hemoglobin will give up oxygen more easily, okay, because that's a situation where more oxygen is needed. So if you look at a plot of the percent saturation of hemoglobin versus the partial pressure of oxygen at different temperatures, at higher temperatures, the hemoglobin, or the oxyhemoglobin dissociation curve, shifts to the right. And that shift to the right is a reflection of the fact that at any one partial pressure, hemoglobin becomes less saturated at those higher temperatures. Okay? And so what that means is hemoglobin gives up more oxygen at higher temperatures, because higher temperatures is a sign that there's a greater demand for oxygen. Okay? So it's kind of neat. Again, hemoglobin is releasing oxygen just at the time when oxygen is most needed. Okay? So the increase in temperature is reflected in a shift of the oxyhemoglobin dissociation curve to the right, or lower temperatures it shifts to the left. Now, a physiologist by the name of Bohr was the one who also first noted that hemoglobin has a decreased affinity for oxygen due to the binding of hydrogen ions to hemoglobin. Now, hydrogen ions load up into the red blood cells when the blood reaches the lungs. Or no, excuse me. When the blood flows into the tissues, usually the concentration of hydrogen ions is higher there, because metabolically active tissues is pretty high producing hydrogen ions. When hydrogen ions bind to hemoglobin, that causes hemoglobin to release the oxygen more easily. Okay, so at low pHs, hemoglobin's affinity for oxygen decreases. At high pHs, it's just the opposite. High pHs, hemoglobin holds on to oxygen more tightly. Okay? So if you look at these series of graphs, again, we have on the vertical axis, we have percent saturation of oxygen. On the horizontal axis at the bottom, we have the partial pressure of oxygen. So when the pH of the blood becomes more acidic, which would be like when our body is more metabolically active, like during exercise, then the oxygen dissociation curve shifts to the right, which means it gives up oxygen more easily, because that's when oxygen is needed the most. Okay, that's called the Bohr effect. So what the Bohr effect means is that at low pHs, the affinity of hemoglobin for oxygen decreases. Okay. Higher pH, the affinity for oxygen increases. All right, now, there's a similar effect that has been noted by a physiologist by the name of Haldane for the affinity of hemoglobin for carbon dioxide. Okay, so when the blood is deoxygenated, like when the blood arrives at our tissues and it gives up oxygen, that increases hemoglobin's affinity for carbon dioxide. So that's reflected in a shift of the carbaminohemoglobin dissociation curve. That's reflected to it. Let's see. This dark blue curve is 100% hemoglobin saturation of CO2, and this light blue one is a percent saturation of CO2 of 0. So when hemoglobin is saturated with oxygen, it holds CO2 less tightly. But when hemoglobin gives up oxygen, its affinity for carbon dioxide increases. Okay, so the Haldane effect has to do with the affinity of hemoglobin for CO2, whereas the Bohr effect talks about the changes in the affinity of hemoglobin for oxygen. Okay, so these two responses of hemoglobin to changes in response to pH changes, and also this Haldane effect is in response to changes in oxygen saturation. Most commonly see questions about these on things like graduate admittance exams, like the MCATs or the advanced GRE test in biology. So for that reason, usually in classes like this, there's at least one question about one or the other of these phenomena on exams, but probably won't be too much. So it's the Bohr effect and Haldane effect are kind of confusing because you can kind of look at it two different ways. You can look at, like, for example, the Bohr effect. You can look at how the affinity for the affinity of hemoglobin for oxygen changes at lower pH, or you could look at it on the flip side, the way in which it changes at higher pHs. So there's, like I said, there's kind of two different ways of looking at it, but they're just opposites. So, okay, any questions? Okay. All right. So another way of summarizing the Haldane effect is the removal of oxygen for hemoglobin increases hemoglobin's affinity for carbon dioxide. And that allows for carbon dioxide to ride on the empty hemoglobin. So one of the ways of looking at this is that when hemoglobin is loaded up with carbon dioxide, there's just not enough room for it to load up oxygen. And so if you get rid of the carbon dioxide, then that makes room for oxygen, and so oxygen can bind more tightly to the hemoglobin. Okay, so you can think of hemoglobin as sort of like a four-seated passenger car, and if it's full of CO2, there's no room for oxygen, so oxygen can't load up on the inside of the car. But if you get rid of some of the carbon dioxide, it can load up a lot easier. All right, now the next part of this chapter is concerned with some respiratory disorders. And there's one type or one kind of category, large category of respiratory disorders that are called COPD. And I found a little video clip on here that I think explains the two different forms of COPD fairly well. So both of these respiratory disorders, by the way, are usually caused by exposure to environmental factors, especially tobacco smoke. Smoking tobacco is the most common factor that can lead to COPD, which is, COPD stands for chronic obstructive pulmonary disease. So with that, I'll let you watch this video.
Chronic obstructive pulmonary disease kills over 100,000 Americans each year, making it the fourth leading cause of death in the United States. So what exactly is chronic obstructive pulmonary disease?
Actually, chronic obstructive pulmonary disease, or COPD, refers to two different diseases of the lung, emphysema and chronic bronchitis. Since emphysema and chronic bronchitis are each usually caused by years of cigarette smoking, afflicted individuals are almost always diagnosed with both at once. Hence the umbrella term chronic obstructive pulmonary disease. This combination of diseases can also be caused by years of inhaling a variety of industrial toxins, fumes, and chemicals. Prolonged exposure to air pollution and secondhand smoke, or even a history of childhood lung infections, can also lead to COPD. However, since chronic bronchitis and emphysema affect two different parts of the lung, let's take a separate look at each of these diseases. Chronic bronchitis is characterized by an inflammation of the bronchial tubes, which are passageways that branch off from the trachea or windpipe and allow air to enter the lungs. This inflammation causes thick mucus to build up in the tubes, preventing sufficient air from reaching the lungs. Emphysema occurs when the walls between the air sacs in the lungs, known as alveoli, lose their elasticity. This, in turn, causes the air sacs to become fragile and overinflated, leading to an unnatural retention of air within the lungs. The symptoms of COPD tend to depend on which disease is more prominent in an afflicted individual. If emphysema is the predominant disease, the first notable symptom will be a shortness of breath during physical exertion. This will soon be followed by a phlegmatic cough and pursed lip breathing. If chronic bronchitis predominates, a persistent mucus-filled cough will often be the first symptom to appear. This may be accompanied by wheezing, shortness of breath, fatigue, and respiratory or lung infections. COPD is usually diagnosed based on an individual's medical history and an examination of his or her lungs. This lung examination can be done with chest x-rays, CAT scans, and any number of pulmonary function tests such as spirometry. A spirometer measures the inhalations and exhalations of the lung and the efficiency of their ability to transfer oxygen to the blood. There is no cure for chronic obstructive pulmonary disease. However, its destructive presence can be controlled and slowed with close medical attention, allowing those afflicted to lead longer and healthier lives. If you are concerned that you may have COPD or are experiencing symptoms of either of its component diseases, please see your doctor or pulmonologist for a checkup immediately.
Now, like I said, the most common cause of COPD is smoking. However, it can also be caused by other environmental factors. Just to give you an example of one of them, my grandmother, on my mother's side of the family, came down with COPD shortly before she passed away from other causes. And her doctors thought that what caused it was that she worked in a shoe factory. She worked in a shoe factory sewing the leather soles on shoes. And when she was working, she breathed in dust from that leather. They were also cutting out the soles of the leather, and that inhalation of that leather dust caused her alveoli to start breaking down, and she came down with emphysema, even though she never smoked a day in her life. So you can get emphysema even if you don't smoke, but smoking definitely increases your chance of getting emphysema or COPD greatly, probably by a factor of 10. So this week in lab, we're going to start the first of 2 lab exercises on the respiratory system. First one we're going to do this week is on spirometry. We're going to learn about how to measure lung volumes, which is one clinical test they can do, pulmonologists can do to determine your respiratory health. And then next week we're going to do a lab exercise on the regulation of breathing.